When you think of pregnancy's unsolved mysteries, you might think of preterm birth or preeclampsia, but you probably don't automatically think of micronutrients. Micronutrients are essential, available in food and supplements, and a rather simple matter, you may think. Actually, micronutrients are kind of like a pregnancy black box, with so little known about how they're metabolized and used in pregnancy that existing dietary intake requirements only tell half the story. The other half has largely remained a mystery. But thanks to my next guest, the secret life of micronutrients and especially iron are slowly coming to the fore, and they're revealing invaluable insights for improved maternal and infant health. This episode is about iron and vitamin D in pregnancy. Stay with us. Joining me today is leading micronutrient expert Dr. Kimberly O'Brien, a professor of human nutrition at Cornell University and the 2025 recipient of the March of Dimes Agnes Higgins Award in Maternal-Fetal Nutrition. Kimberly, welcome to the show.
Thank you for having me.
Congratulations on being this year's Agnes Higgins Award recipient.
Oh, thank you very much. I'm so honored and humbled to receive this award.
Well, anyone who's ever talked to you would believe that you are honored and humbled because you are so passionate about this topic. And when I say this topic, I mean micronutrient metabolism, micronutrient use, and requirements in pregnancy. How did you get here? How did you become so interested in micronutrients and during gestation?
Right. So I started working with pregnant adolescents in Baltimore. And at the same time I was working with calcium in the pregnant adolescents, I was working with pregnant women living in a slum in Peru. And 50% of the Peruvian women were anemic. It looked like we were having even more anemia in our pregnant adolescents. So we went through 10 years of medical records and we discovered that 60% of our pregnant adolescents were anemic, even though many of them were overweight or obese at entry into prenatal care, and even though they had access to prenatal supplements, whereas the Peruvian women did not. And that just really shocked me. Women living in our country receiving the standard of medical care were so much more anemic than women that were living in an under-resourced area. And it really just hooked me on trying to understand what was it about pregnancy in a young gravida that was different than an older woman. And I just became very interested in iron and maternal fetal partitioning.
Yeah, wow, that is fascinating. I want to know why even, well, I know a little bit about that because I interviewed you before and the answer is that regardless of how much we take in, only some is absorbed and used by our bodies, right? So, and you're going to talk a lot about that during this episode, and that's kind of the crux of the episode is what do we know about what happens once it's ingested, and
Right.
so little is actually used. So we are going to dedicate most of this episode to iron, like you said. You fell in love with that micronutrient a little bit. So tell me why this micronutrient is important in general. You know, for folks that maybe there's a woman that doesn't want to get pregnant. Why is it important for women? Why is it important for men, for children? And why is it important for women that are pregnant and babies? And maybe talk a little bit about anemia here, like both the Peruvian and the American populations that you worked with.
Right. I think one of the reasons that iron is such an interesting nutrient is the only nutrient that we can't physiologically excrete once we absorb it. So once we let it into our body, we only get rid of it really small losses every day. But if we get too much, we can't eliminate the excess iron. So our body has to very tightly regulate the amount of iron that comes in. And that's unique compared to other nutrients. Because we can't get rid of it, we're not that efficient at letting it in. We have to very tightly regulate the amount that we get in relation to what our body needs. And that requires a lot of different mechanisms in order for us to do that effectively. Why we need that iron is we need iron to make hemoglobin. Hemoglobin is the protein that moves oxygen throughout our body. If you don't have enough iron, you can't make enough hemoglobin, you don't deliver enough oxygen to your tissues, and that has a whole host of adverse problems. And so one of the things that is unique about pregnancy is during pregnancy, We need a lot more iron. We need about 1,000 milligrams of iron to support a typical singleton pregnancy. And of that 1,000 milligrams, 300 goes to the baby, and that's what the baby has at birth. Normally, we only absorb 1 to 2 milligrams per day. So you can see how much higher that load of iron that we need to support pregnancy is in relation to what we normally absorb in a non-pregnant individual. So while all age groups require iron and need it for very important functions, it's particularly important during pregnancy because the very large amount of iron that pregnant individuals need to expand their blood volume, to make the placenta function properly, and to give that 300 milligrams of iron to the newborn so that it has enough iron to get it through that first 6 months of life.
Do all people, you just said that pregnant women need more than other populations. Do other populations in general need more or less the same amount? Do each of us as individuals vary in the amount that we need? And is it possible to have a genetic predisposition for iron deficiency? Some folks that just are always low, no matter the supplements, no matter the diet.
Right. So that's a really interesting question. And a lot of the nutrition field is moving towards precision nutrition metrics to try and identify who might need more, who might need less. And that's a very complicated but very important question. Not many people have genetic disorders that make them at risk for iron deficiency. Those genetic disorders exist, but fortunately, they're relatively rare. So if you are to provide iron to an individual from the diet or from the supplements, most people should be able to absorb that iron. But as I said before, because we don't want to let too much in, we don't absorb a lot of it. So you have to have kind of a continual presence of iron for a longer period of time in order for you to compensate for a deficit. It's not something that we just can tank up on very quickly because we absorb 100% of it.
Right. So let's get into that exactly then. So what do we know about how much dietary and supplemental iron is absorbed into the body in general, I guess, in pregnancy, let's say that, and during non-pregnancy? How much, where do we get our iron from, and how much that we take in, whether through pills or food, is absorbed and used?
Right. So we get iron in our diets from animal protein as heme iron, and we get iron as inorganic iron, the type of iron we get from plants or supplements.
Are both important?
Both are very important. Both of those are sources of iron. What's interesting is that heme iron is much better absorbed, and we don't regulate it very tightly in relation to our body iron stores. We also don't really know the protein that we use to absorb heme iron, so there's still a lot of questions. The prenatal iron the kind of iron that we get in supplements is inorganic iron. That's a very complicated form of iron. It is very tightly regulated, and we use many hormones in our body regulate that form of iron where they don't regulate the heme iron. So we get iron from lots of different foods, both animal foods, plant foods. We can absorb iron from both of those forms. And our body is trying to regulate that using the inorganic iron, using hormones. So we have a hormone called hepcidin in our body that we make. We make more of it as we get more iron in our body. And both pregnant and non-pregnant individuals can regulate how much iron they absorb with that very important iron regulatory hormone. As you have more iron in your body, you make more hepcidin, and that prevents us from absorbing iron or releasing it from stores. So there's two other very important hormones that our body also uses to control this. So we do have the capacity to very tightly regulate non-heme iron absorption from our diet and from supplements. And those pathways are very similar between pregnant and non-pregnant women. The one interesting component to that is some research in animals suggests that pregnant animals can downregulate that hepcidin hormone early in pregnancy, independently of iron status. Whether or not that happens in humans, we don't really know the answer to that question yet. It's harder to start these studies very early in pregnancy, as many women don't know they're pregnant yet, and they're not enrolling in research studies at those very early stages of pregnancy.
We'll talk about some of your research a little bit later on, but I would very uneducatedly guess that maybe humans don't possess the ability to downregulate that hepcidin because of how many women are anemic at delivery. Your research has shown, right? Because if they had the ability to downregulate, then more iron would be able to get absorbed. They wouldn't be anemic. So I don't. Maybe there's a
Yeah.
link there. But Kimberly, tell me then what percent of iron is actually absorbed compared to what we take in?
Right. So on average, kind of the estimates are if you have a moderate bioavailability diet that has a mixture of plant and animal foods, you
Sure.
absorb about 18% of the iron in your diet.
One
That's not,
eight. Wow.
yeah, one
Yeah.
eight, 18%. But we only need 1 to 2 milligrams of iron per day to maintain balance if you're not pregnant. Where this gets complicated is during late gestation, you can need up to 8 milligrams of iron per day to meet your needs. And that's
Mm.
why prenatal supplements contain 27 milligrams of iron to really help provide enough iron so that absorbing a relatively small fraction of that iron is going to help you meet those high iron demands of late pregnancy.
And much of your work is focused around finding out exactly how much iron is absorbed, where does it go, who needs it, how is it used. And the way that you do this is very scientific. It is one of this process is one of the things that sets you apart from other researchers in your field. And this is the use of tracer techniques to safely trace the movement of these micronutrients in the body. This allows you to see how much of the micronutrients we eat are absorbed and used by the body. Can you please explain how tracing works?
Right. So many minerals that we eat in our diet exist in different weights, and some of those forms are very rare. So what we do in our approach is that we purchase highly enriched forms of the rare iron that we eat in our diet, and then we can measure those stable isotopes with special instrumentation called a mass spectrometer that allows us to differentiate all of the different forms of iron that we get in our diet. Then by feeding this special tracer to someone, we're able to measure it in their red blood cells. We can measure it in their placenta. We can measure it in their baby at birth. And we can get a lot more detailed information about how that individual is absorbing iron, what factors are associated with iron absorption. And I think most interestingly, how much of that absorbed iron does the mother use versus the placenta versus the fetus?
Right. And this was one of the most interesting things that I learned from you when I interviewed you for the press release that we wrote about your win, which is that there is a competition, an intense competition happening between the mother, the baby, and the placenta. They're not all working harmoniously together. They're working for themselves in the
Mhm.
battle for iron. Tell us a little bit about what's happening there.
Yeah, so all three of those individuals or entities need iron in order to survive, right? And so it's not just an easy answer to tell you that all individuals do the same kind of partitioning. There are many other factors that are going to influence where that iron goes. And we're still trying to figure out what are those important determinants of how much the placenta gets versus the baby versus the mother. Using these isotopes, we can actually try and differentiate that. And we have found different answers to that question if we're working in a biologically mature mother or a biologically immature adolescent. There are differences in where that iron goes and whose needs predominate. And the things that we find in humans are not always the things that are found in animal models. Animal models tend to suggest that the placenta is selfish. All of the iron is first prioritized to the placenta. The placenta has kind of a constant level of iron, even if you make the mother very iron deficient. We have found in our adolescent and adult studies that it's not as clean as that. Between a biologically immature mom and a mature mom, there seems to be something about the young mother that helps prioritize iron for her use, whereas an older mother tends to be better about prioritizing iron more going towards the baby. So I think we don't have one easy answer to say that all individuals when they're pregnant are going to put X percent here, X percent in the placenta, and X percent to their baby. And as we'll talk later on, there's also differences even how the baby can address that iron and what is unique about the fetus and how it's able to pull iron across the placenta.
Absolutely. So let's talk about the young mother versus the older mother then. Does a woman's iron, this might be a silly question, but do the iron stores grow as a woman ages? Because you would think they would, otherwise why would the body of a younger mother protect her over her baby essentially and the placenta. So
Right.
tell me about what we know about that.
Yeah. So a young mother is still growing. She's still
Yes.
laying down muscle mass. She still has growth.
And
And I think from,
can we define young mother? What
yeah,
are we
so
talking here?
a young mother is someone that would get pregnant soon after their menses begin. So
So
this, we have done these
13.
studies, and young mothers can be 11, they can be 12. So you can get pregnant when you're still growing, when you have not finished your linear growth, when you have not fully consolidated your muscle mass. So you still need those nutrients to support your own development. And that's where I think you have this different kind of competition going on that may kind of prioritize those nutrients for the biologically immature mom. Animal models have found that if you do studies in adolescent sheep, they use those nutrients for themselves more than sending them to their baby. I think from an evolutionary perspective, that makes sense, right? Because the mother can have many reproductive cycles, but you would want her to kind of enter that process as fit as she can be so that she can have many of those reproductive cycles. And there may be something that is unique to the biological immaturity. There may also be something that's unique with the first time you make a placenta, but it seems to be kind of driven more by the age of the mom than just being a first pregnancy, because older individuals are having babies at later and later ages. And you really see this in the individuals that are still growing.
So does that mean that an 11-year-old needs more iron than a 35-year-old during
In,
pregnancy?
in general, right now, the kind of requirements, an 11-year-old does need more iron than a mature individual because they are still laying down tissue. So
Hmm.
iron requirements and many nutrient requirements are higher when you're growing so that you can support the amount of nutrient that's needed to build your body mass.
Yeah. Interesting. And when does that end? We know that the brain stops developing around 25. When does our body stop building blood mass and volume and tissue and all that?
Yeah. It depends. Like, so if you're talking about your skeleton, most women by the time they're 17 have achieved 95 or so percent of their adult skeletal mass. There are small increments till they're 30, but they're much, much smaller than what you see by the time you're 17. You get most of your bone mass by the time you're 17.
Hmm.
For iron, it's just a function of muscle mass and blood volume. So as you continue to grow, you have a larger blood volume, you need more hemoglobin, you have more iron. As you build muscle mass, that's another depot, a body store of iron that you also need to put iron into. So it's not like it's difficult to kind of give a finite answer of by the time we're 21, we're done. It depends on how you're growing.
Yeah.
But by the end of adolescence, in general, you've really made most of the increases you're going to have in your body compartments that require more nutrient to form them.
Hmm. We've all heard about teenage pregnancy. Obviously, that's a concern in this country and other countries as well. Is it happening as young as 11 years old here, or is that kind of the outlier? That seems to
That's
me extraordinarily young.
Yeah, that's an outlier, but it can happen, right? You can get pregnant as soon as your menses begin, and the
Yes.
age at that happens can be as young as 11. Fortunately, it's not commonplace, right? It's
Hmm.
possible, but more adolescents becoming pregnant are 15 to 17 than you
Yeah.
see under the age of 15, but it
Hmm.
does happen.
Hmm. So we talked about the battle between the placenta, the baby, and the mom. There is another battle being waged in the womb, and that is between the fetuses that are in there at the same time when we're talking
Mhm.
about women that are having multiples rather than singletons. What determines the victor here? You would think that all of these fetuses are the same biologically,
Right.
right? Because they're twins or triplets. And then you think about, and do each of them have their own placenta, Kimberly?
Yeah, so that's an interesting, sometimes yes, sometimes no. So
Really?
you can have multiple fetuses in the same placenta, in the same amniotic sac. So you can, it's called monochorionic momo twinning. So you can have, they can share a placenta, they can share an amniotic sac, or they can have their own. So there's all different permutations of, or combinations. They can be the same sex, different sex, but they don't all have an equal amount of iron. And you can even visualize that if you just look at the birth weights of multiple birth babies. Sometimes you have discordant growth where siblings will weigh more than 15 to 25% difference in birth weight. So they clearly grew differently in utero, even though they had access to the same maternal nutrition, the same blood supply. And I think a lot of this difference is driven by the placenta. So the placenta is the conduit for all nutrients that fetus is going to acquire over gestation. And that placenta may form more or less efficiently. And there's something that's very interesting, even if you look at the baby's sex. A male fetus makes a small placenta but weighs more at birth compared to a female fetus. A female fetus makes a big placenta but weighs less at birth. So it's said that the male placenta is more efficient than the female's placenta. No one knows why that early stages of placental formation, something differs between male and female fetuses that allow that early process to differentiate. Where that becomes important is if the woman has a nutritional insult over pregnancy, that smaller placenta is less capable of sustaining the fetus. And it's why you see the sex ratio differs if you have something like a famine or something that really is a large nutritional insult to the mother. The females tend to be born at higher percentages than males because they're less capable of surviving those kind of nutritional deficits in utero. You see the same thing in multiple births. There's more females than males. So there's a whole host of different combinations that influence the efficiency of nutrients getting to the fetus. And I think a lot of them are driven by the placenta.
Well, isn't that fascinating? So the boys, the males are more efficient at kind of efficient at what? Efficient
at getting more nutrients can be transferred across a smaller placenta, so that less of the investment
basically
goes
getting
into
more nutrients to them.
Yeah, and making placenta a smaller placenta. So they don't put as much of their energy into building a big placenta. They make a smaller placenta, and then that smaller placenta maybe has to work harder because they end up larger. So you make ratios between the birth weight and the placenta weight, and you can use that as a metric of how efficient that placenta was. And there are differences by sex.
The males are more efficient at getting the nutrients to them with a small placenta, but it's also their curse in a way when it comes to these, like you said, an insult during pregnancy, like a mass one, like a famine or something. So there's two sides of every coin.
Right.
That's absolutely fascinating. So one of the other things that you've been doing during your career in this field is trying to find the best biomarker for iron deficiency in moms and babies. So what do we know today about the best biomarker and whether it's translatable? You and I had a conversation about you think you found the best one or the best combination of hormones, I believe, but that maybe it's not something that we can bring into the clinic. Tell me what we know about that, and where does ferritin stand in this whole conversation?
Yeah, so there are a lot of iron status biomarkers, and ferritin is one of them. And ferritin is a good biomarker. The only problem with ferritin is that when you have inflammation, that makes ferritin get higher because ferritin is an acute phase protein. So you may see a high ferritin and attribute it to a high iron stores, but it's actually high because that woman has inflammation. We're getting better at making normative equations that we can use to measure an inflammatory marker and ferritin to account for that, but it's still not perfect because of that inflammation biomarker interaction. When we have done our research, we've measured ferritin, we've measured inflammatory biomarkers, and we've measured the three important hormones that our body uses to regulate iron homeostasis. And from all of those markers, we found that it was the ratio between erythropoietin and hepcidin that was the most predictive for identifying women that went on to develop iron deficiency or iron deficiency anemia. Why we think that is that erythropoietin senses hypoxia. When the mother doesn't have enough oxygen, you make more EPO. And hepcidin is a measure of iron deficiency. When you don't have iron, you don't have a lot of hepcidin. So that ratio together is allowing us to measure both hypoxia and iron deficiency. And we believe that's why it was a better biomarker. You asked is that clinically relevant? Probably not very useful because both of those markers are relatively expensive compared to many other iron status biomarkers. Many hospitals don't have panels to order those. And there's not good standards of at this level, this is your risk, right? They're research tools, very important research tools that help us figure out how iron is controlled and regulated, but they're unfortunately probably not going to have much clinical utility at this point in time.
How early in pregnancy did you take them in your work to determine that those were good indicators of risk?
Right. So that's a really good question. In our studies,
And if I may, what percentage of women that showed a risk increase actually went on to develop anemia or high levels of these hormones?
yeah, so I don't recall exactly the
But a significant amount.
yeah, it was highly predictive. So it was a
Okay.
very strong biomarker compared to
Okay.
the others. How early can we get these measures? That's another very important question. So many times we're not able to really start getting blood samples from women until about 12 to 14 weeks of pregnancy. So they're in their second trimester. Women often don't enter. They all don't enter early. They don't realize they're pregnant. When they first go into prenatal care, there are a lot of other tests that are being done, right? So it's hard to enroll a pregnant individual into a research study very early in pregnancy because of all of the other dynamic changes that are going on and care changes that are happening. In our studies, we do get measures in the first trimester, but they're at the latter stage of the first trimester, closer to 12 weeks, and we get measures longitudinally. So these measures earlier in pregnancy, some women are already anemic or already have iron deficiency anemia in the first trimester. Others don't develop it into the third. So these are all just nuances of when is the nutritional deficit present. It may be present before pregnancy. It may be developed in the first, second, or third trimester. And all of those scenarios are somewhat different. And it also depends on how likely are you to get these early blood samples, which is often quite challenging during pregnancy in a research study.
You mentioned a little earlier that babies also have a superpower, and that is that they can regulate the production of hepcidin, which is one of the most important iron hormones. Why is it important? Does that mean that a baby can essentially say, I need more iron, so I'm going to lower the amount of hepcidin in my system that is being signaled in my mother's blood? And so then the gates can go down and more of the iron that she eats will get absorbed and come to me.
Yes, exactly. So that is what we found. So we tried to ask that question using a multiple birth model. So we had a pregnant woman carrying twins, triplets, or quadruplets. So we had one fixed maternal environment and then up to 4 neonates at birth. And what we found is that hepcidin was more variable between the siblings than it was between unrelated neonates. So the baby can sense the iron that it gets, and its liver makes hepcidin in response to how the internal iron environment of that fetus. We also found that the hepcidin that the fetus was making was significantly correlated with a very important protein in the placenta that the placenta uses to move iron out of the placenta to the baby. So animal studies find some, not all animal studies find that same association. So this is what we found in our multiple birth model. It really does look like the baby can sense its own iron stores. Those iron stores are going to vary between different fetuses in the same uterine cavity based on how efficient their placenta is at delivering that iron to the baby, or maybe how big the placenta is, or how well expressed the proteins are that are needed to move iron to the baby. But the baby does have a capacity to regulate its own production of hormones in relation to its internal iron status.
And was this very surprising?
Yeah, we didn't think we would see so much variability within, the same mother, the same blood supply, the same uterine cavity, but there was a lot of variability. I'm not sure why it surprised us because, as I mentioned earlier, you can see huge variability in weight. You can have siblings that are vastly different in how large they are at birth, which tells you that something is different about how effective they are at stealing nutrients or getting nutrients from the mother.
Yeah. Silly question. What does iron and oxygen have to do with birth weight?
Yeah, so the baby needs oxygen for all of its metabolic functions, right?
Okay. So
it's
just the foundational at the very bottom. Development ways, in order to get development, you must have
Yeah.
oxygen, you must have iron. Okay.
Right.
Growth.
Iron's really
Growth.
important for mitochondria too. Mitochondria generate energy. So you need iron not only to make hemoglobin, but you need iron for mitochondria to function properly. You need iron to make other very important proteins and enzymes that the body uses. So it's one marker, not the only marker. You make an important point that, it's a combination of nutrients that are necessary to support robust growth in a fetus. A combination of having those nutrients available, but also having a healthy functioning placenta that has the capacity to move things to the fetus in an optimal way.
Mm-hmm. So we're talking about babies. Let's keep talking about babies. Do we know what constitutes normal iron levels in a newborn? Or is that a question we don't have the answer to?
Right. That seems like a question we should have the answer to, but we do not have the answer to that question, in my opinion. We do have the answer to that question for hemoglobin. There's a lot of normative data on hemoglobin. You can measure hemoglobin in a newborn, and you can very accurately determine whether or not that neonate has anemia. What's less certain is how do you interpret iron status biomarkers in a baby at birth? In utero, the baby makes a lot of hemoglobin so that it can effectively scavenge oxygen from the mother. Once the baby is born, it starts breathing air. It doesn't need that high hemoglobin anymore, and it rapidly breaks the hemoglobin down. And then because the iron can't be excreted, that iron gets redistributed into different iron storage proteins. And all of these things are going on over the first few weeks of life. What we don't have is a lot of normative data to look at how those things are changing over the first few weeks of life. No mother wants their baby to be stuck multiple times so that we can figure out what's going on with these iron status biomarkers. And even things like hemoglobin are not routinely measured at birth. We don't measure whether or not a newborn has anemia unless they have risk factors like preterm birth or low birth weight. So that's another really, I think, a gap in the literature. We really don't screen for anemia at birth because it's thought that most babies are going to have enough iron. And when we're measuring anemia, we're measuring the really extreme deficiency. We're not capturing babies that might not be anemic, but they might not have enough iron stores. And iron stores are really important over early development to support the rapid growth that babies have over the first 6 months of life.
Related to that, Kimberly, you found that some of your most kind of prominent research has found that there is a high prevalence of anemia among high-risk pregnant populations, specifically teenage mothers, we talked about them, and women carrying multiples. You found that despite receiving iron from dietary and supplemental sources, up to 40% of these women and up to a quarter of their babies were anemic at delivery. So it sounds to me like, and it sounds like what you're saying is that we should be screening babies at birth. Is that what you're saying? These are granted, this is a high risk population, but
Right.
it sounds like maybe there's some other women that are falling through the cracks here, and that we should be screening their babies not only for anemia, but like you said, for optimal health. Maybe not anemia, but other an almost anemia or halfway there to anemia. Conditions that could have negative impacts.
Right. I really do think we need to be screening. We screen babies for a lot of very rare metabolic diseases, but we don't screen for anemia. So everyone, it's quite common that I get the argument that we see this because we work with high risk pregnancies. We work with teens, we work with multiples, and that we would not find this level of anemia in otherwise healthy women. Recently, we went through all of the medical records at the University of Rochester over the past 10 years. We pulled out over 40,000 women's medical records, and we found that a quarter of these women had anemia by late gestation. So while it was up to 40% in these higher risk populations, a quarter of women being anemic by late gestation when they don't have other medical complications, I believe is concerning. What we
Yes, that's
can't see,
25%.
yes, that's 25%. What we can't see from the medical records is we don't know what impact that has on their baby because we don't screen babies. So we don't know how much iron the neonate at birth has. And that, I think, is a big gap that we need to start addressing and looking at pregnancy and nutrition as a dyad. We want to make sure that the woman has enough iron to prevent anemia, but we also want to make sure that the woman has enough iron so that she's giving that fetus as much iron as she possibly can so that it's born with an optimal amount of reserve. that's going to get it over those first 6 months of life. Right
Yeah.
now, we don't coordinate those two individuals well enough when we're thinking about nutritional demands.
Would it make more sense to screen women in late pregnancy for anemia and then screen their babies, the women that show that they're at risk, or to screen all babies?
Yeah, I think until we know a lot more about what's happening with iron stores at birth, we need to kind of screen all babies, and then we know what the norms are and what the extremes are. Why, whether or not we should just start screening in the third trimester, I think that one of the important things about pregnancy is so much is going on at the very earliest stages of gestation when the placenta is being formed by the fetus. And we want to make sure that at that point in time, there's enough nutrients around to really allow that process to occur at an optimal level. So it's really, I think that what we actually need to do is make sure that individual has enough nutrition, optimal amount of nutrition before they become pregnant, so that they enter pregnancy with iron stores in a non-anemic state, and so that all of these processes can continue, not worrying about not having enough of a nutrient that you need to start partitioning it in ways between those three entities that might not be optimal.
Right. Absolutely. Totally agree. But then there's still the added kind of what if in late pregnancy when the pregnant mother needs more, even, right?
Yes.
When it jumps up.
Yeah. And this is a whole other area of research to how do you replete them quickly? So if you identify them in late pregnancy as being very low or iron deficient or anemic, we don't absorb a lot of iron. So we only have a few weeks left before that woman's going to give birth. So there's a whole body of work now that's looking at, do you give women intravenous iron? And does that allow them to really get a fast repletion at a very crucial life stage, as opposed to just adding more iron into the diet when we know that you don't absorb a lot of it and it's going to take you some period of time to really replete your stores at a time when we know the fetus also is sucking up 8 milligrams of iron per day at the high end. So it's hard to just intervene effectively and efficiently in the latter weeks of pregnancy.
So you bypass that barrier process of iron if you go straight to the IV?
Yes. Yeah, you
Okay.
bypass the gut. And so you don't need to worry about hepcidin blocking it from being absorbed. You just deliver it directly into the blood.
Mm-hmm. So obviously we don't have normative levels for babies at birth. We don't exactly know how much pregnant women need. We know that they probably need more than they're getting, though, right?
Yes.
Because there are a quarter of women in the low risk population that are anemic at delivery, unbeknownst to them or the OBs that were treating them at the time. It looks like, and a quarter of them, and 40% of high risk populations anemic, and a quarter of their babies. So it seems like it's not a controversial thing to say that we should be getting more iron in pregnancy. Uh, but the only way we could have the evidence to prove that is through a randomized control trial, and that will not happen due to ethical concerns associated with conducting this work on pregnant women. So where do we go from here? Where do you go from here? Late at night when you're thinking about your work and kind of the future of it, and how to make it translatable and clinically relevant, and how to actually help these women and prevent these worst case outcomes. What are the thoughts that you think, Kimberly?
Right. I think it's kind of, I think from many different levels we have to approach this. I think we need better research funding for these kind of normative questions, right? These are basic questions. What level of biomarker is optimal for health outcomes? Those are basic questions. We don't have enough research funding really to carry that out. So barring a lot more research funding to address these kind of basic questions, I think we need to better capitalize on the medical records that we do have from very large number of women. All women generally get hemoglobin measured over gestation. Some of them get ferritin measured, but we don't take advantage of the medical records and mine the medical records in an efficient or systematic way nationally, so that we can actually figure out what fraction of our population has hemoglobin that's suboptimal across gestation. I think it would be nice to be able to screen newborns at birth for hemoglobin. We screen them for the many rare diseases, as I mentioned, but this becomes a healthcare cost issue. You know, if the health system is constrained, if we don't have a lot of resources, and if there's a perception that babies are going to be born iron replete, then that's not a question that's prioritized. So I think we have to draw attention to the risk that exists. I think we have to find more research funding to answer these kind of questions. And I think we need to capitalize on resources that we do have access to that are going to give us more information, like the medical records.
You often talk about there's a difference between optimal health and iron deficiency, and you want to focus on bridging that gap a little bit. So talk to me about that.
Yeah, so our nutrient requirements are defined as the level of nutrient needed to prevent deficiency. So for iron, what is the level of iron needed to prevent anemia? It's not the level of iron needed to ensure that the woman still retains some iron stores and still has a healthy level of hemoglobin across gestation. It's
so
not
you could be in trouble without being deficient, is what you're saying.
Exactly. You could be iron deficient without anemia. Is that optimal? I would argue no, that's not optimal. You could also, you're defining a level of a nutrient during pregnancy that's only the level needed for the mother. That might not be the level needed to fully endow her fetus. We need to think of that as a dyad. It is a dyad. There's two people nutritional requirements during pregnancy. We need to consider what is the level of nutritional intake to fully meet both of those individuals' demands, and also to make sure that the placenta is going to function optimally. And right now, we're only focusing on biomarkers in the mother, not what is present in the newborn at birth. So I think that that's something that makes it harder to identify what is an optimal level of intake. An optimal level of intake really isn't the level of intake needed to prevent deficiency, but to promote health. And in general, all of our nutritional requirements now are really defined to prevent deficiency, not promote optimal health. So I think that that is one of the key problems in how we undertake these studies.
What symptoms, or maybe if iron deficiency without actually qualifying as anemia for a pregnant mother or a baby at birth, if it is asymptomatic, perhaps it is, what does that look like, either symptomatically or asymptomatically inside the body, not having anemia but being iron deficient?
Yeah, if you're iron deficient, you may not be as energetic. I mean, you don't see frank consequences of a nutrient till you do reach the deficiency state. So if you're truly iron deficient, you may not have enough energy. Your work performance may go down. In a newborn, there may be cognitive deficits. If you don't fully myelinate your brain while you're developing, it's a process that needs iron that may have long-term irreversible consequences on the subsequent newborn. For women that during pregnancy, looking at anemia, you see that's associated with low birth weight and preterm birth. It's harder to associate iron deficiency with those really extreme outcomes. It may be that iron status is associated with depression, and there are other outcomes that are important to consider in brain function. We need iron for many different processes in our body, not just producing hemoglobin. It's just harder to find those links and to look at it. Another challenge in the nutrition field in general, a woman that's iron deficient may be also deficient in several other nutrients. And it's hard to disentangle the relative role of each of those nutrients and how they're impacting outcomes. That's especially true for things like iron that we ingest. High-rich animal products contain a lot of iron, but they also contain a lot of zinc. So often individuals have multiple micronutrient deficiencies, and it makes it a bit harder to attribute outcomes to any one given nutrient, particularly if you can't do a randomized controlled trial and give a lot of that nutrient to one group of women and withhold it from another.
I just have one last question on iron. It's a specific question about the differences between plant and animal iron. So heme and non-heme, right? You mentioned in the beginning that our body needs both. That means that vegetarians are not getting something that they need. Is that right? And folks that only eat meat and don't have a plant-based diet are not getting a source of iron that they need. So tell me a little bit about that. And then you also mentioned that, I believe you said that heme iron, so animal based iron, is better absorbed by the body. Did you say that?
If you do eat a mixed diet containing meat, you absorb more of that heme iron. So you can subsist just perfectly fine on a plant-based diet, but because you don't absorb as much iron from the plant sources, you have to be more careful about
I
what you're
see.
ingesting and making sure, that you're monitoring your intake. There are other nuances to that question too, because we don't yet know how iron that we're eating influences our gut microbiome.
Mm,
Some bacteria in our gut need heme. Some bacteria, they can't make it themselves. They're reliant on us eating it. And we don't yet know
mm.
enough about how iron in our diet is shaping our gut microbiome. So there are a lot of unanswered questions about the relative form of iron that we might be eating and some other effects that it may be having in our body that may or may not be optimal, right? So we can get by just fine with only plant-based iron. That's fine. It's just they are absorbed different. They are regulated differently. We absorb about 50% of the heme iron, but we only absorb about a far lower fraction in the tens, like 10 to 18% of non-heme iron, depending on what kind of plant source it has and how many other inhibitors or enhancers you have in your diet.
And that 18% figure, that was in pregnancy?
That 18% was pregnancy in women that had a mixed diet of
Okay.
moderate bioavailability, so a
Okay.
mixture of meat and plant sources.
So it seems like in pregnancy, you're absorbing even less than you are when you're not pregnant, because you said heme is 50% absorbed, and then plant is between 10 and 15, and then you get to 18 in pregnancy. What's happening there?
Yeah, a lot of that data, that those recommendations were made some time ago. They haven't been
Mm,
updated in a while, so that's another
okay.
limitation. So it's hard to pick. That's the number they used. There are other data that have been published since then that might change some of those numbers a little
Okay.
bit.
Yeah.
So you what the important point is, you do regulate your absorption in relation to your status. Both pregnant and non-pregnant individuals, when you do not have enough iron in your body, your absorption goes up. So humans are pretty good about adjusting the amount of iron they absorb from their diet in relation to their body iron stores. Both pregnant and non-pregnant women, if they don't have iron stores, they're going to increase the efficiency of iron absorption. Both men, women, pregnant, non-pregnant. We all have the same regulatory system, with the caveat being that some people think that there's something unique about pregnancy that allows them to dampen down that hepcidin early in pregnancy to maybe give them an edge and help them better get iron in during pregnancy.
Well, so that's good news, right? Our bodies are doing their jobs. They're doing an incredible job at it. It's our job now to take it in, right? The
Yes,
body
yeah, eat a good diet.
will exactly. The body will open the gates, but our diet, our supplements and our diet, and we need to know that we need that our bodies are, they will do their job, but they can only absorb so much, which means we have to continue to ingest.
Yeah.
All right, let's talk about a little bit about vitamin D before we conclude our recording here. So, your team recently developed the first tracer method to follow the absorption and metabolism of vitamin D, the same thing that you did with iron in the pregnant population. What did you find here with vitamin D, Kimberly?
Yeah, so we were able to label vitamin D with a stable isotope. We fed it to pregnant and non-pregnant women, and then we measured how that D was absorbed and how it was further metabolized into other metabolites along the pathway to make the vitamin D hormone. What we found is that one of the most significant determinants of vitamin D half-life, which is what we are measuring, is the vitamin D binding protein. And so that in both pregnant and non-pregnant women, the vitamin D binding protein concentration was a very important determinant of the half-life. We are in the process of extending that study now. So that was our pilot data. We've received more new grants so that we can now look to see how does vitamin D metabolism vary between women that have a lean body mass or that have excess body adiposity. And we're looking at the role of vitamin D binding protein genotype, because there are different forms of vitamin D binding protein depending on your genotype. and some of those don't bind vitamin D as strongly. So what we're hypothesizing is that women that have a lot of body fat, they're known to have a greater risk of vitamin D insufficiency. It may be because vitamin D is sequestered in body fat. Vitamin D is a lipid-soluble vitamin. Or it may be that the obesity itself alters some of the enzymes that are needed to activate vitamin D. So we're going to be looking at that in our study. We're focusing on individuals that have a greater risk of vitamin D deficiency. Individuals with darker skin tones are at greater risk of vitamin D deficiency because darker skin tones, it takes longer to make vitamin D in sunlight when you have darker skin tones. Individuals with darker skin tones also tend to have a vitamin D binding protein that doesn't bind vitamin D as avidly, so that it's cleared faster from our body. So we're hoping in this new study that's going to recruit non-pregnant and pregnant women, lean women and women with obesity, and darker skinned individuals and lighter skinned individuals, that we're going to get a better understanding of what are the factors that are influencing vitamin D dynamics in women. This also, we're going to be focusing on the baby. The baby doesn't get sunlight in utero. It's 100% dependent on the mother's vitamin D status. So we're going to be looking at vitamin D status in the baby. We're going to be looking at vitamin D metabolism in the placenta. And we're hoping just to get a more comprehensive understanding, like we've been doing for iron, about the factors that are influencing how much vitamin D is partitioned between the mom, baby, and placenta.
Wow, that seems like a big study, and fantastic that you're that you're getting, pregnant, not pregnant, darker skinned, lighter skinned, obese, not obese. So a lot of data will come out of that. Last question for you, Kimberly. You half joked to me during the interview that we had when I first got to know you that you, and I quote, stalk the L&D floor, that's the labor and delivery floor at your local hospital in Ithaca, looking for samples from new mothers to check what form of vitamin D is present in the placenta. Okay, so several questions here. So are you looking for placental samples? Can you just take these samples from women that have delivered? And why does and of course, the most important question for you, why does the form of vitamin D matter? Are there different forms for different women? Is it one form of vitamin D? Do we didn't know the form before this? I figured we'd know the form. Tell me what you're doing at the hospital, Kimberly,
Yeah.
and how often do you go there? Do the nurses know your name?
These studies that you're referring to, we do in Rochester. So I work with women in Rochester in the large obstetrics clinic. Okay. We're always looking for placentas. We'll never turn a placenta away. We have freezers full of them. There are endless questions, and we collect them, as many as we can get our hands on. What we were doing in the labor and delivery floors, we wanted to see what form of vitamin D crosses a placenta. And the data on this is mixed. We also don't know what are the factors that determine the relative amount of vitamin D that goes to the fetus. And we were trying to do these studies and not being able to trace the tracer if we gave it to women earlier in pregnancy. So what we thought is, we'll wait till a woman is in active labor. We'll go and we'll ask her if she's willing to drink this tracer, and then she can drink it, and then she delivers her baby shortly thereafter, and we can see if that vitamin D was transferred to the newborn. And we gave her different forms of vitamin D. The vitamin D that you get in your supplement is usually vitamin D3, but that gets metabolized into our body into something called 25-hydroxyvitamin D. That form might cross, either one of those forms might cross. And so we used tracer labeled D3 and tracer labeled 25-hydroxyvitamin D3, fed it to the woman, took cord blood at birth. Unfortunately, there were some contaminating components in the cord blood, so we weren't able to answer that question yet. We now have developed better resolution mass spectrometric techniques that we think we're going to be able to resolve those contaminant peaks, and we'll be able to answer that. We should know what form crosses, but we really don't know what form crosses. And a tracer is a perfect way to ask that. By labeling something, we can see, does that label get over into the newborn?
So why does the form matter, whether it's D3 or the other one that you mentioned? And out of curiosity, what were the contaminants? Why were you not able to see the form?
Yeah. So we don't exactly know what the contaminants were. One of the limitations with a mass spec is it separates things according to mass. So it was something that had a very similar mass to vitamin D that we
Mm,
weren't able to resolve or separate apart enough so that we could quantify what we needed to. And as mass spectrometers get more and more as they evolve, they have a better capacity to resolve adjacent peaks that are very similar in mass. So now with new instrumentation, we think we can address this. Why it matters, there's a lot of questions about the form of vitamin D that you can give someone. There are studies that give 25 hydroxy vitamin D to humans because you absorb it better. So individuals that have chronic kidney disease can be treated with 25 hydroxy vitamin D. It's not what's normally in a prenatal supplement, but if we were to find that that form of vitamin D is much better absorbed and perhaps much better able to cross the placenta, maybe there's some utility in dosing with that amount. Again, for the same reason, if you have a population that's D deficient, maybe using a form that you absorb better and transfer better would give you a better opportunity to replete a fetus. So it's important just from a scientific question to try and understand what are the forms that cross, how does the placenta metabolize those forms, and then what is found in the baby at birth. It's still not really known.
yeah. You're answering so many questions that you would think we had the answers to, and we don't. And you are looking to uncover them all. I thank you for that.
Well, thank you. It's fun.
Fantastic. It has been an honor, Kimberly. Thank you for your work. It's so important. You're a tireless crusader and a true champion for maternal and infant health, which is why March of Dimes gave you the Agnes Higgins Award in Maternal-Fetal Nutrition. You care about this stuff deeply. You're extraordinarily bright and smart, and it's been an honor to interview you today. So thank you for coming on the show.
Thank you very much for having me. It was really a pleasure.